Hello, hi, a very good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So yes, Satyendra, hello, a very happy good evening. So guys, please do like, share and subscribe the session. For all those who are new, Monday, Wednesday and Friday I teach a class 12 chapter, and Tuesday, Thursday, Saturday I teach for a class 11 chapter. So that's really nice. So we were done with almost 5 chapters in human physiology. So today we are entering into a new chapter, that is neural control and coordination. The first part of which is the human nervous system. So today we'll talk about the structure of neuron, and before that, how is the CNS organized? So what are the different nerves here? So all that is what we're going to talk about. Alright, yes, I hope your locomotion and movement is clear. So Satyendra, yes, I saw your question about stretch receptors, right? That is micturition. So let me explain. So stretch receptors, basically the function of stretch receptors is to take the signal from the CNS. But one more thing is that your stretch receptors also make space in the urinary bladder so that the urine can be stored inside the urinary bladder. So if at all there is no stretch receptors, no one can hold the urine there, and it will be a continuous process. So micturition will happen. So micturition is just excretion of urine. So micturition will happen, but it will no longer be under the voluntary control. So if the stretch receptors are absent, so it will no longer be a voluntary control, but the excretion or the release of urine will happen. Alright, yes? Okay, so Priyam, yes, good evening. So thank you so much for joining my session. So I guess you're new to my session, so please do like, share and subscribe. Shimpi, hello, yes, very happy good evening. I am fine, how are you? So Priyadarshini, hi. Okay, alright. So I guess I have answered your question, Satyendra. So now moving forward, so before we actually move into this topic of neural control and coordination, so I have an announcement. So today once again we are meeting live at 7 p.m. So we are going to talk about the quick recap of the cell cycle and cell division. Actually this we did it in the last class, but we didn't complete the entire revision, right? We only completed mitosis. We still have to talk about your meiosis portion. So this is part 2 of your cell cycle and cell division. Alright, so here we are going to talk about your meiosis. Fine. So here we'll talk about meiosis. So this session is at 7 p.m. today. So please do join me live for this session. So along with meiosis, I will also discuss some numericals from this topic, that is cell cycle and cell division. Alright, yes. So now shall we talk about your neural control and coordination. So this is lecture 1. So we'll talk about your neural control and coordination. So what are the topics that are included in this chapter? So we shall see it here. So the neural system, so the human neural system. So how is the neural system organized from lower vertebrates to higher vertebrates? And then we'll talk about the human neural system, then neuron, the structure and functional unit of your nervous system. Then we'll talk about your central nervous system. Then under your central nervous system, we'll talk about your brain in detail. So then we'll talk about your reflex action and your reflex arc. Alright. Then sensory reception and processing. So under the sensory reception and processing, basically we are highlighting your sense organs. So under that we'll talk in detail about your structure of your eye, and then followed by the mechanism of vision. Okay, then human hear, and we will talk about the mechanism of hearing as well. Okay, so these are the topics for this chapter. Okay. Okay, so now, just like we justified why do we need a locomotory system, why do animals locomote, so why do we have to excrete substances from your body, why do we have to digest your food, why do we have to have circulatory system, so similarly why should you have a nervous system, and if at all we have a nervous system, why should there be something called as coordination? So without coordination, can't our body organs survive? Can't it work properly? Can't it work smoothly? So before that, let me define what is coordination for you. So coordination is the process through which two or more organs interact, okay, and complement the function of one another. So imagine one day your digestive cells, the cells of the digestion process, say the cells of small intestine says, I am not going to perform any digestion, no conversion of sucrose into glucose, etc., etc., no breakdown of proteins, but I will do the function of your heart. And if your heart says no, I will only do, then there's going to be a chaos, right? So one main unicellular, one main very, very interesting feature or the most beautiful characteristic of a multicellular organism is that we work in an organized manner. So multicellular organisms work in the organized manner so that you have division of labor, right? So when I say there is division of labor, which means one organ has to interact with the other and it has to coordinate, right? Yes, Antonia B, hello, hi, good evening, a very, very happy good evening. Okay, so this is called as coordination, fine. For example, if this girl is jogging right now, so if she is jogging, then her muscles need more energy. If her muscles, for the muscle contraction, which we spoke about in the previous chapter, muscle contraction requires ATP. So if your body has to produce more ATP, then your body needs more oxygen. If your body needs more oxygen, then it has to breathe high, right? So it has to increase its rate of inspiration. So even if it inhales oxygen, your blood has to be oxygenated, right? So first increased rate of respiration to take in more oxygen, then increased heartbeat, okay, then increased blood flow via blood vessels, because oxygenated blood has to flow inside your muscles, right? So just for this one activity, that is jogging, so your respiratory system has to work, your circulatory system, that is your heart has to pump pump blood, and your circulatory system, which are part of your your blood vessels, which are part of your circulatory system, they also have to come into picture. So just for one activity, you need the coordination of your respiratory system and your circulatory system. Thus, now read the definition. So coordination is the process through which two or more organisms, they interact or complement the function of one another. All right? Yes? Okay. Now talking about neural system. So the neural system of all animals is composed of highly specialized cells called as neurons. So just like you had sarcomeres in your muscles, nephrons in your kidney, so in your neural system you have your neurons. Fine? Okay, so what are the functions of neurons? So your neurons detect, receive and transmit different kinds of stimuli. So that is the function of a neuron. So your neurons will detect, receive and transmit different kinds of stimuli. Okay, so for one example is your hydra. So your hydra belongs to your lower animals. So it comes under your animal kingdom. So under your phylum Coelenterata. So your hydra has a network of neurons. Okay, so though it is a very lower animal present in the lower phylum, it has a network of neurons. Alright, so in case of insects, so moving further higher up the phylum, that is your insects which belongs to phylum Orthopoda, in that case your brain, it has a brain, it has a number of ganglia and neural tissues. So this is how the organization happens from the lower to the higher hierarchy. So in case of your excretory system also we saw, right? So in case of Platyhelminthes, it was your flame cells. In case of Ascihelminthes, it was excretory pore. Yes, so as we go up the hierarchy, the organs are getting more complicated. So is the case here. In case of phylum Orthopoda, so the insects have a brain, the number of ganglia and neural tissues. Alright. Yes, Harini, very, very happy good evening. So please do like, share and subscribe. So thank you for joining us. So vertebrates have well-developed neural system. So in case of lower phyla, so we just have a network of neurons. In a much higher phyla, you have brain, ganglia and neural tissues. But as we go much higher up the hierarchy, under phylum Chordata, subphylum Vertebrata, we have well-developed neural system. Okay, fine. So this is how your hydra looks like. So it has a network of neurons. So going more further, so now talking about the human neural system. So the human neural system has two parts. One is central nervous system, the other one is peripheral nervous system. Okay, so the CNS, you know, it consists of your brain and spinal cord. So PNS talks about all nerves of the body that is associated with your CNS. So when I talk about the central nervous system, so when I talk about the human nervous system, it is of two parts, central nervous system and peripheral nervous system. So central nervous system consists of brain and spinal cord, and your peripheral nervous system consists of all the nerves of the body associated with your CNS. Okay, fine. So moving further, so now we will talk about your CNS. So the whole nervous system is derived from the embryonic ectoderm. So your embryonic germ layer has 3 layers of cells: ectoderm, endoderm and meso. So ectoderm, mesoderm and endoderm, right? So your heart is of the mesodermal origin, if you remember. So here your nervous system is from the ectodermal origin. So please do not forget the origin of the nervous system. It is ectodermal origin. So your CNS consists of your brain and spinal cord, alright. So they are formed from the neural tube that develops from the ectoderm after the gastrula stage of the embryo. So right after fertilization, your zygote undergoes a series of divisions, a series of cleavage to form blastocyst. So that is 8 to 16 cell division, right? So in that stage, your neural tube is formed from the ectoderm of the germ layer. Alright, so it develops from the neural tube in the intrauterine life. So within the uterus, everything happens, that is the development of the neural tube. Okay, fine. So the anterior part of this neural tube, if this is my neural tube, the anterior part develops into the brain, and the tail portion, the caudal, is the posterior part, it develops into the spinal cord. Alright, yes. So this is how it is. So you have your neural tube developed into spinal cord as well as into the brain. So the anterior region develops into brain and the posterior region develops into the spinal cord. Fine. So here you have your forebrain. So this is in the womb position. So this is in the development of the embryo. So this is in the intrauterine condition. So your neural tube, the anterior part develops into the brain and the caudal part develops into the spinal cord. Alright, yes. Okay, so moving further, so talking about your PNS. So your PNS is the peripheral nervous system, which talks about all the nerves associated with your CNS. Okay, so the nerve fibers of the PNS are of two types. One is afferent nerve fiber, the other one is efferent. So where else have we read about afferent and efferent? In your glomerulus, right? So the blood bringing, so the blood vessel which carries blood into the glomerulus is called as afferent. The one that is taking blood out of the glomerulus is called as efferent, right? So similarly here, so the nerve fibers of PNS are of two types, afferent and efferent. Alright, so afferent is from the organ to the CNS. Efferent is from the CNS to other organs. So to make it pretty clear, let me write here. So PNS has two types of nerve fibers. The first one is afferent nerve fiber. Okay, the next one is efferent nerve fibers. Okay, so your afferent nerve fiber carries information from your target organ to CNS. In case of your efferent neuron, it goes from your CNS into your target organ. Since here the information is moving from the target organ into the CNS, it is called as your sensory neuron. Since the information is passed from the target organ to the CNS, so your afferent nerve fibers are also known as sensory neurons. Alright, so here since the information goes from the CNS to the target organ, it is known as a motor neuron. So if you remember when we were talking about the junction of muscles, so the neuron carrying the information and the muscle fiber waiting to receive the information, it was called as a motor unit, right? So which literally means the information or the neuron carrying the information from the CNS to the target organ is your motor neuron or your efferent nerve fiber. Alright, is this much clear? So you understood what is efferent, what is efferent, and afferent is also known as your sensory neuron, efferent is also known as your motor neuron. Fine? Okay, so moving forward, your PNS also has further classification. It is classified into somatic and autonomic neural system. Okay, so your autonomic nervous system has further two types, that is sympathetic and parasympathetic. So apart from these two, you also have another type, that is visceral. So visceral nervous system is also a part of your PNS, where the whole complex of nerves, fibers, ganglia and plexus are being supplied. Alright, so let me write it like this once again. So your PNS has two types of fibers, so which we just saw. One is your afferent nerve fiber, so which is also known as your sensory neurons. Okay, so the next one is known as your efferent nerve fiber. Okay, so this is known as, okay, you are taking notes, alright, so this is known as motor neurons. Fine, so this is only talking about the nerves of your PNS. Apart from that, PNS is also divided into somatic and autonomous. So this is somatic nervous system and the other one is autonomous nervous system. Fine, so somatic means when the nerve fibers are connected to the skeletal system. So when the nerve fibers are connected with the skeletal system of the body. Okay, so that is, so when it carries information to the skeletal system. So in case of your skeletal somatic nervous system, so the information flows from your CNS to your skeletal system. Alright, yes. So in case of autonomous, it goes to your involuntary organs. Involuntary or hollow organs. So here the information moves from your CNS to your involuntary organs. Alright, so since here in both the cases the information is going from the CNS, so both your somatic and autonomic neural system, it consists of your efferent nerve fibers or your motor neurons. So here, because here in case of your somatic nervous system also, the information is going from your CNS to the skeletal system. Here also from CNS to the involuntary. So both are your efferent nerve fibers or your motor neurons. Alright, so did you get this? Yes? Okay. So now going further into this. So your autonomic nervous system is further divided into two types. Okay. The first one is sympathetic nerve fibers and the other one is parasympathetic. Fine. So this is the classification of your PNS. So Antonia B, am I fine? So should I repeat any point? So Shimpi, is it clear? Yes. So you're understanding what I'm teaching? So you understood the classification of PNS. So PNS, the nerves are divided into afferent and efferent, and your PNS is also further classified into your somatic neural system and your autonomic neural system. So in case of somatic, the information is going from the CNS into the skeletal system. So here it is going from the CNS into the involuntary organs. Alright, your autonomous is further divided into sympathetic and parasympathetic nervous system. Clear? So this sympathetic and parasympathetic neural system are always opposite in action. If this sympathetic increases your heartbeat, parasympathetic will decrease your heartbeat. So this is one example. So your sympathetic and parasympathetic always work antagonistic to each other. So antagonistic means oppositely. They work antagonistically to each other. Alright, yes, so shall I go forward? Okay, so moving further, so let's talk about your neurons. So talking further about your PNS, so this I've already told you, your PNS transmit impulses from the tissues or organs to the CNS, that is your afferent fiber. So this is also known as your sensory nerve fibers. Okay. So your efferent fibers, they carry information from the CNS to the target organs. So they are known as motor nerve fibers. So here is an image, alright. So moving further. So your somatic neural system, so it relays information from the CNS to the skeletal system. So once again, here also you have efferent nerve fibers or your motor neurons present here. So autonomic nervous system or the neural system, this transmit impulse from the CNS to the involuntary organs or the visceral organs. Fine. So now talking about the structure of a neuron. So now so long we were talking about how your nervous system is divided. So into CNS and into PNS. So your CNS was neatly classified into brain and spinal cord, whereas your PNS had many categories based on the nerve fibers and based on where the information is reaching, right? So now talking about the neuron. Okay, so what is a neuron? So like I told you, neuron is a structure and functional unit of your neural tissue. Fine? So this is the structure of a neuron. So now we are going to talk about the human nervous system. So now I am going to describe the structure of neurons. So please make sure you understand each and every step. Okay. So the structure of a neuron, when I say we are going to talk about the structure of a neuron, remember there are three main structures of the neuron. Okay. So here we go. So we are going to talk about the structure of the neuron. There are three main parts. So first part is called as the cyton or the cell body. Fine. So the next part of the neuron is called as your dendrites. Then the third one is called as an axon. So these are the three main parts of a neuron. So first one is a cell body, which is here. The second one is dendrites, which is here. The third one is axon. So there are three main parts of a neuron. Now explaining each of them. So cyton means a cell. So it is also known as a cell body. So when I say it is a cell, so it is going to have cytoplasm. It will also have a nucleus with it. So can you see the cell body has a nucleus. It also has a cytoplasm. Apart from that, it has minute granules which are present in the cell body, which are known as Nissel's granules. So your cyton, which is the cell body, has a cytoplasm, has a nucleus. It also has a Nissel's granules. Fine. So the next part of your neuron is a dendrite. So dendrites are short, repeatedly branched filaments just near to your cell body. Okay. So they are short and repeatedly branched structures, and your dendrites also contain your Nissel's granules. So dendrites also contain your Nissel's granules. So these short filamentous but branched structures which are very near to your cell body are called as dendrites. Alright, clear? The third one is an axon. So axon is a long unbranched filaments. Fine. So this long line is your axon. So you have three parts when you talk about the structure of a neuron. So the first one is the cell body, which consists of your nucleus and your cytoplasm. The next one is your your dendrites. Then you have your axon. Alright. So the portion on the cell body from where your axon arises. So once again, the portion on the cell body from where your axon arises. So this portion is known as axon hillock. So this portion is known as axon hillock. So the end of the axon is known as axon terminal. So this region is known as an axon terminal. An axon terminal ends in what is known as a synaptic knob. A synaptic knob is a sac-like structure. Okay, so these round, so they are called the synaptic knob or synaptic vesicles. So they contain your neurotransmitters. So in the previous chapter, when we were talking about the mechanism of muscle contraction, I told you a neuron from the brain brings the signal to the muscle. So when it reaches the muscle fiber, it releases your neurotransmitters, right? So your neurotransmitters are released from the synaptic knob. Alright, is this much clear? Okay. So your axon is surrounded by a fatty sheath layer. So your axon at regular intervals, it is surrounded by a fatty sheath layer. So this is known as myelin sheath. So myelin sheath prevents the leakage of energy during the conduction of impulse within the axon. So the information has to pass throughout the length of the axon, right? So in between, no energy should leak outside. So that is why it is covered with a fatty sheath that is called as myelin sheath. So who secretes this myelin sheath? You have a specific group of cells which are called as schwann cells who secrete your myelin sheath. So schwann cells are present. Can you see these black cells? The schwann cells are present which secrete your myelin sheath. So in the cytoplasm of the schwann cell, you have your myelin sheath. But remember, your myelin sheath is not a continuous one. So it is like this. If this is my axon, so my myelin sheath is like this. Okay, so it is not a continuous one. So the gap where your myelin sheath is absent, it is called as your node of Ranvier. It is called as the node of Ranvier. Alright, is this much clear? So this is how you will describe the structure of a neuron. So when I say we are going to talk about the structure of a neuron, we have three main parts: cyton or the cell body, the next one is your dendrite, and third one is your axon. Alright, yes? So your cyton or the cell body talks about your cytoplasm nucleus, and it also has your Nissel's granules. Then we are talking about your dendrite. So your dendrites are short, repeatedly branched sequences or filaments which also possess your Nissel's granule. Okay. Then axon is a long unbranched filament. So the portion of the cell body from where your axon arises, that is known as axon hillock. And your axon always terminates in your axon terminal, which has a synaptic knob, and synaptic knob contains neurotransmitters. Fine. So your axon is also covered by a fatty sheath which is known as myelin sheath at regular intervals, and these myelin sheath are secreted by schwann cells. The portion on the axon where your myelin sheath is absent, it is called as node of Ranvier. Is this much clear to everyone? Yes. Any doubts? Anthony or B? Yes. Do you have any doubts? Should I repeat any point here? So let me know if I have to repeat any point. So things are clear to everyone? Yes. So shall I go move forward? Okay. Anthony or B, is it clear? So should I repeat anything? So Satyendra, any doubt? Shimpi, are you watching? So Priya, are you also making notes? So there was someone called as Priyam. Are you also there? So if I have to repeat, if you didn't understand, so please stop me then and there. Okay? Okay. So Anthony says clear. So I am moving forward. Okay. So this is whatever I have explained. It is in your theory. So we spoke about your axon hillock, that is here. So some axon give rise to side branches which are called as collateral fibers. Okay, so I told you your axon is not branched, but sometimes if it's branched, it gives rise to collateral fibers. So just like your muscle fiber had a plasma membrane, your axon also has a plasma membrane which is known as axolemma. So in case of muscle, it was called as sarcolemma, right? The cytoplasm is called as axoplasm. So now to distinguish it clearly, so we are talking about a muscle here, and we are talking about a nerve. Okay, so in a normal cell, so you call what is a plasma membrane. So in the muscle, the plasma membrane is called as sarcolemma. So in a neuron, it is called as axolemma. Fine. So you have cytoplasm in your cell. The cytoplasm of a muscle is known as sarcoplasm. Right? And the cytoplasm of a neuron is known as axoplasm. So now you should not get confused between sarcolemma, axolemma, sarcoplasm and axoplasm. In case of muscle, you have another special organelle which is known as sarcoplasmic reticulum, which is very homologous to your endoplasmic reticulum. Alright, yes, are these terms clear? What is a sarcolemma? What is an axolemma? Then what is sarcoplasm? What is a sarcoplasm, axoplasm and sarcoplasmic reticulum. Okay, yes, fine. Now axon ends in a structure that is called as synaptic knob. So this also I have told you. Then synaptic knob comes very close to the dendrites of the next neuron to form what is called as a synapse. So let me show you here. So this is the rough structure of your neuron. Okay. So let me draw your neurons roughly like this. So this is your dendrites, right? So which are short and repeatedly branched. So this is your cell body. So you have an axon. So axon terminates in your synaptic knob like this. So now immediately the next neuron starts and this continues. So in between the two neurons, you have a space that is called a synapse. So the junction between two neurons is called a synapse, and sometimes here you have a very minute gap. So you have a microscopic gap of 200 angstroms, which is called as your synaptic cleft. Alright, so this junction between two neurons is called as a synapse, and there you have a microscopic gap which is 200 angstrom, which is known as your synaptic cleft. Alright, fine. So moving forward, so based on the number of axon and dendrites, your neurons are divided into three types. What are the three types of neurons? Multipolar neurons, bipolar neurons and unipolar neurons. So on what basis they are categorized? Based on the number of axon and dendrites they have, your neurons can be of multipolar, bipolar or unipolar, right? So now we'll talk about what is multipolar. So from the term multi is many, right? So they have one axon but two or more dendrites. They have one long unbranched filament but they have many dendrites. So that is multipolar condition, and multipolar condition is seen in your cerebral cortex. Alright, so now here you will remember the types of neurons as well as the examples associated with it. So multipolar, multi is many, so you have one axon but two or more dendrites. So such a kind of neuron is called as multipolar neurons, and it is seen in the cerebral cortex of your forebrain. So cerebral cortex is present in your forebrain region. Fine? Okay. The next one is bipolar. So bi is two. So they have one axon, one dendrite. So only two regions, and it is found in the retina of the eye. Alright. So the third one is unipolar. So uni is only one. So the cell body with only one axon is present in your entire neuron, and such a type of unipolar neuron is seen in the embryonic stages. Okay. So once again, based on the number of axon and dendrites, your neurons can be divided into your multipolar neurons, bipolar neurons and unipolar neurons. Alright, so your multipolar neurons is where you have one axon and one dendrite, one axon and many dendrite, and it is seen in the cerebral cortex. So cerebral cortex is a portion of your forebrain, okay, the front part of the brain. So then bipolar, so bi is two, so you have one axon, one dendrite. It is found in the retina of your eye. Fine. Then unipolar is one, so you have only one axon along with the cell body, and it is found in your embryonic stages. Alright, yes, so shall I move forward? So yes, so this is your bipolar, unipolar and multipolar. Okay? Okay. So now talking about the nerve fibers, that is about the myelin sheath, okay. So an axon of a neuron is covered with 2 or more sheaths, okay. So on the basis of presence or absence of sheath, your nerve fibers can be of 2 types. One is medullated or myelinated nerve fiber, the other one is non-medullary nerve fiber, okay. So now talking about your medullated or myelinated nerve fiber. So around the nerve, so you have a sheath or a fatty substance that is formed, okay. So thus it is called as a medullary sheath or myelin sheath. So around the nerve, you have a sheath of fatty substance that is formed. So this is called as a medullary sheath or your myelinated sheath. So the medullarated sheath is not continuous, and the point of absence of your medullary sheath is known as the nodes of Ranvier. So I told you, no, if this is your axon, so you have myelin sheath like this. The point of access of your myelin sheath is known as your nodes of Ranvier. Alright, is this clear? Okay. So the part of the medullated nerve fiber between 2 adjacent nodes is called as an internode. So this is node of Ranvier. So again here it is a node of Ranvier. So between 2 nodes of Ranvier you have an internode. Alright, clear? Okay, so the medullary sheath, it basically forms an insulating coat, and what is the function of this myelinated sheath? It prevents the loss of energy during the conduction of nerve impulse. Alright, so the medullary sheath plus the node of Ranvier surrounded by a transparent cellular outer covering is the neurolemma of the squam cell. So squam cell is one particular type of cell present in the axon of your neuron, and in the cytoplasm of the squam cell you have your medullary sheath and you have your nodes of Ranvier arising from. Alright, is this clear? So there is always this question, who secretes your myelin sheath? So it is your squam cells. Alright, fine. So shall I move forward? So Anthony Abhi, are you making notes? So should I repeat anything again? So is this clear to everyone? Yes? No doubts? So shall I go forward? Yes, Anthony Abhi, things are fine. So this can seem as a big topic, but it is actually not. If you understand the logic, it is very, very simple. So should I repeat any point? Come on, quick, let me know. Clear. Anthony Abhi says clear. Fine, okay. So all the others who are watching, if I have to repeat something, so do let me know. So moving forward, generation and conduction of nerve impulse. Okay. So Babu, Baya, so hello, so I guess you're new to my session, so thank you so much for joining my session. So please do like, share and subscribe. So I will talk about generation and conduction of nerve impulse. So now there are 2 terms here. What is conduction of impulse and what is transmission of impulse? So conduction of impulse is when the impulse moves along the length of the axon, and transmission of impulse is how the impulse is being passed on between 2 neurons. Okay, so when the impulse has to be passed from one neuron to the other, we call it as transmission of impulse. When the impulse has to be passed within the length of the axon, it is called as conduction of impulse. Alright, yes? Okay, so now let me write it here. So now this is our axon. Okay, so I am not drawing a cyton, I am not drawing, okay, so let me draw, it's not tough. So this is our structure of a neuron. So now here we are going to talk about the conduction of nerve impulse. So conduction means that is transfer of impulse along the length of the axon. Okay, so how it is going to happen? For that you should understand, so this is the axoplasm, right? So this is called as the axoplasm. So the axoplasm or the membrane of the axon is polarized. So point number 1, to understand how conduction of impulse happens within a neuron, you should understand that the membrane of the axon is polarized. So what do I mean by polarized? Which means it is charged. How does it get charged? How does it obtain charge? How does the membrane become charged? Because it has a lot of ion channels present on its surface. So these ion channels are selectively permeable to certain kinds of ions. So once again, we are going to talk about conduction of impulse. So conduction means movement of impulse within the axon. Alright, so for that you should understand the membrane of the neuron or the axon is polarized. So what do I mean by polarized? It is being charged. Alright, so how it is getting charged? From the ion channels which are present on its membrane. Yes, so these ion channels are selectively permeable to some ions and they are impermeable to certain ions. Alright, so your axonal membrane, it is very permeable to K+ ions and negatively charged proteins. So, if your axonal membrane or your axoplasm is very permeable to your K+ ions and negatively charged proteins, thus on the inner side of your axoplasm, so you have a lot of K+ ions plus negatively charged proteins like this. Okay, but on the outer side of your axoplasm, you have more concentration of Na+ and positively charged proteins. Fine. So on the outer side, you have Na+ and positively charged proteins. So now on the inner side you have negative charge and on the outer side you have positive charge, right? So thus there is a difference between the charge. So this is called as gradient. Thus an ionic gradient is set up here. So inside of the membrane you have negative charge, on the outer side you have positive charge. So thus point number 4, an ionic gradient is maintained. An ionic gradient is set up. So what enables the formation of the ionic gradient is the presence of your sodium-potassium ion channels. It is only because the presence of these ion channels on the membrane of your neuron, this ionic gradient, this difference in charge is established. Alright, so because of this ion channel. So this ion channel forms a policy of 3 Na+ moving out of the cell to 2 K+ moving inside the cell. Okay, moving into the cell. Fine. So this is how your sodium-potassium ion channel works. So did you understand this much? So we are talking about the conduction of nerve impulse. For the conduction of nerve impulse, you should understand the membrane of the axon is polarized, meaning it is charged because it has ion channels. So the inside of the membrane or the inner region of the membrane is permeable to K+ and negatively charged proteins. Alright, so thus on the interior you have more of K+ ions and a lot of negatively charged proteins. On the outer side you have lot of Na+ ions and positively charged proteins. Thus, an ionic gradient is set up because you have negative charge inside, positive charge outside, right? Thus, an ionic gradient is set up. So, this ionic gradient is made possible by the presence of sodium-potassium ion channel on the membrane of your axon. Alright, so this follows a criteria of 3 Na+ moving out of the cell and 2 Na+ moving into the cell. Now, is this clear? Yes? Fine. So, now pay attention. Once again, so this is my axon. So I am dividing my axon into parts. Okay. So this is my A part, B part and C part. So when the neuronal membrane is resting, when it is not conducting any impulse, so it is going to have positive charge on the outer side and negative charge on the inner side. So when the axonal membrane is at rest, okay, so when the axonal membrane is at rest, so it will have negative charge on the inner side and positive charge on the outer side. So this potential difference on the neuroplasm at the resting stage is known as your resting potential. So the potential difference across the neuroplasm at a resting stage is called as the resting potential of the plasma membrane. So the resting potential is around -70 millivolts. So, hello Vinu, hi, good evening. It's okay, but please do watch the previous part of the lecture as well so that you understand this concept. It is a very important concept. So, when the neuron is resting, when it is not conducting any impulse, it has negative charge on the outside and negative charge on the inside and positive charge on the outside. Alright. So, this potential difference across the resting plasma membrane of your neuron is known as resting potential, and the value is minus 70 millivolts. Okay. So, now pay attention. If at all a stimulus is applied to this region of A, now what will happen? The membrane becomes permeable to Na+. So, suddenly if there is a stimulus that is applied to this portion, then the membrane of the neuron becomes suddenly permeable to Na+, and Na+ moves into the cell. So, when Na+ moves into the cell, what will happen is that more amount of positive ions enter inside the cell. Okay? Yes? So, when the neuron was resting, it had negative charge inside, but when a stimulus is applied, the membrane permeability changes and it becomes permeable to Na+. So, now the inside of the membrane becomes positively charged. Thus, here the membranes are polarized, they are charged, but here you have reversal of charges. So, this reversal of charges is known as depolarization. So, now your axonal membrane is depolarized. Alright, yes, but this membrane permeability of Na+ is very short-lived. So, understand. So, this action potential across the plasma membrane is known as the nerve impulse. So, one more point. So, here I told you what is a resting potential. Now, I am telling you what is an action potential. So, this potential difference along the length of a neuron, when a particular stimulus is applied, that is called as an action potential or a nerve impulse. Alright, but this membrane permeability of Na+ is short-lived. So, to quickly restore the initial equilibrium, your K+ from the inside moves out. So, now your K+ is moving out. So, if your K+ moves out, so what will happen? So, immediately it comes back to this stage that your outside concentration is positive and inside concentration becomes negative once again. Alright. So, now once again, initially it was like this. Then when an impulse is applied, it becomes reversal. The charge is reversed, it becomes depolarized. Now, after the impulse is getting transmitted inside, it once again becomes repolarized. It will once again get back to this position. So, understand, now it has an impulse being acted upon. So, the charge is getting reversed on the inside and the outside by changing the permeability of the membrane to Na+. But this membrane permeability to Na+ is very short-lived. So, to restore the resting potential, your K+ moves out and once again it becomes repolarized. Alright. So, on the inside, the impulse moves from A to B and on the outside, it moves from B to A. Alright. So, this is called as conduction of nerve impulse. So, did you understand what is conduction of nerve impulse? Vinu, Anthony, yes. So, Babu, did you also understand? So, is this clear? So, I will stop here. So, in the next class, I will once again give a recap of this and then talk about the transmission of impulse. So, transmission is how the impulse is getting passed on between two neurons. Okay? Yes? So, I hope you understood whatever I taught. So, don't forget, we are meeting once again for a live session at 7 p.m. So, where we are going to talk about meiosis. Okay, so this is what I told you. So, whatever is told here, I have told it in the figure. So, this is what happens. So, suddenly there is a induced membrane permeability to Na+. So, Na+ moves in. So, the charge gets reversed. So, that is called as depolarization. Alright. So, in the next class, we will talk about the transmission of impulse. Okay, fine. So I hope you understood whatever I taught. So please don't forget my live session at 7 p.m. So thank you for being a wonderful audience, and I'm waiting to meet you at 7 p.m. today. So if at all you have any doubts, so once again get back to me. So I'm here to always ready to clear your doubts. Okay. So have a great evening. So looking forward to meet you again. So thank you and stay tuned. Bye bye.